Right, now I'm going to show you how to do transcription first. And this table here is not used for transcription. It's going to come up in just a minute. So first we just have to think about the DNA, and when the double helix separates, this is the strand that RNA polymerase will read. So given that DNA, what letters of RNA do I want in this row here? And so that will be the smaller copy of the gene that will leave the nucleus and be translated next. So for the first two codons here, I'll just show you how I would do that. The first DNA sequence is AAG, and for that I have to add the complementary RNA. So A goes with T in DNA, but if you watch the video, you'll remember that in RNA we have U instead. So that will be uracil, uracil, and then G corresponds to C, UUC. Now I've transcribed the first codon. Click into the second one and do the same thing. So C transcribes to G, A transcribes to U, and RNA still has adenine, so T will transcribe to A. Now I have the first two codons transcribed. After transcription, and I would recommend you just do them all the way across, and then do translation next. After transcription, I have to think about how to transcribe that into amino acids, translate it, sorry, into amino acids, which is, pretty different. And in the cell, that is done with the help of ribosomes and transfer RNA, and it happens outside the nucleus. This genetic code table here reflects the result of a lot of research where scientists would, for example, the first one, put together a whole bunch of mRNA that was nothing but uracil, and then put that in a test tube with some ribosomes and some transfer RNA and all the different amino acids, and what they got was nothing but phenylalanine when that was translated. All right. So with a whole long series of those types of experiments, scientists discovered this genetic code, and this table shows you what the ribosomes do when they read different kinds of mRNA. Our mRNA was UUC here. So if we look in the code table, the first letter is U, and the second letter is U. So we're up in this quadrant here, and if we find UUC, that encodes Phe, or phenylalanine. So I'll just type P. Sorry, I've got caps lock on. Capital P H E there for phenylalanine. The next mRNA codon is GUA, and I can find that right down here, GUA, and that encodes VAL, or valine, VAL. And so that's all there is to it. You start by using the complementary bases to make your mRNA, with the only catch being that instead of T, RNA has U. Fill all those in, and then use the genetic code table to fill in the amino acids. The middle set of amino acids here were just provided for you, so you wouldn't have to do this sort of tedious procedure for all 31 codons in the list. Now let's scroll down and look briefly at this mutation section. And one thing that I just want to clear up is that there are a few of the codons that are filled in completely for you, and that's because there are no mutations in those, and so they're going to stay the same, and this represents normal human insulin. And then these are grayed out because we're going to explore what would happen if there were different DNA sequences here that would then affect the RNA and potentially the amino acids at each of those places. So I don't expect you to type anything into these cells here. They're just there for reference. What you need to do is go down to this row, this table here, and read what it says. So in codon 11, what would happen if the DNA, instead of being AAC, was AAG? The normal human insulin is AAC. What if it changed to AAG? There may be somebody out there in the world that has that sequence in their DNA. That would change the mRNA. So normally AAC is going to be, what is it, G. And if it's AAG, now it's going to be UUC instead. Oh, that should still be purple. I'm going to have to change that. Oh, wow. Okay, so now we look at the genetic code table again. We say normally the mRNA is UUG. That's what would be right here in the normal insulin. So let's go see what that would give you using the code. UUG is leucine. So that's the normal, so we type that one in here. Leu, the normal amino acid, changes to something else potentially. UUC. What does UUC encode? UUC encodes Phe, or phenylalanine. So when you're done, it should read Leu changes to Phe. And that's an example of one mutation to just one letter in the DNA that then affects the identity of just one of the amino acids in the insulin protein. What you're going to do is go through and do that same thing with the rest of these, and you'll have to do codon 11 too. So I showed you the answer, but it'll help you practice just to do it yourself. And then answer the questions referencing these examples. So seeing what happens in each of these cases, what are some things that can happen to a protein if you change just one letter in the DNA? And I hope you learn from that and enjoy it. These are made up examples here, but the mutations in this initial table are real research-based examples of mutations that do occur in insulin genes in some people. And very often those people are born with unusual kind of neonatal type of diabetes where they're diabetic from before birth. And most of us have a different type of diabetes. As you'll learn later, there are a lot of other ways that insulin can be disrupted. But this is just an example highlighting some of the inherited forms of diabetes as a way to start thinking about mutations and how they change DNA and how they can change proteins. So I hope you enjoy. Thanks.